let's start with the first part, which focuses on centrosomes and centrioles, as both are complicated cell structures that are essential for cell division. Okay, basically, I will explain about the centrosome cycle, which consists of duplication of centrosome and the separation of centrosome. This process begins at the same time as DNA replication, which is triggered by G1S CDK and SCDK. At G1 phase, cells have a single centrosome containing a pair of centrioles called as parental centrioles, which embedded in the pericentriolar material. Next, during S phase, centrosomes remain together while duplication of centriole occurs, and it's marked by the formation of procentrioles at the end of each parental centrioles. Move to the G2 phase, procentrioles elongate until they reach the length of the parental centrioles. Lastly, during early M phase, the centrosomes separate and each nucleates its own ester of microtubules, and it will form opposite poles of the bipolar mitotic spindle.
Let's talk about microtubule dynamics during cell division, but first we will discuss about the structure of microtubules. So, microtubules are composed of protein called tubulin dimer that are arranged one after another to form a chain. Now this structure has structural polarity, and this refers to the two different ends, -N and a +N. -N stay anchored in the centrosome. On the other hand, the +Ns are dynamically unstable, where they continuously switch back and forth between polymerization and depolymerization by the addition and loss of their tubulin subunits. This occurs in a process called dynamic instability. As the cell enters mitosis, the rapidly growing and shrinking microtubules extend in all directions from two centrosomes, exploring the interior of the cell. To prevent from disassembling and depolymerization, the +N of microtubules must look for an attachment to be stabilized. As a result, some of the microtubules grow from one centrosome and interact with the microtubules from other centrosomes in the presence of microtubule-associated protein and motoproteins, which cross-link the microtubules together in an overlap zone indicated in black dots. This helps stabilize the +Ns by decreasing the probability of their depolymerization. The two centrosomes that give rise to these microtubules are now called spindle poles, whereas the interacting microtubules are called interpolar microtubules. This simple strategy of random and selective stabilization forms the basic framework of the mitotic spindle with its characteristic bipolar shape.
The mitotic spindle begins to form in prophase. This assembly of the highly dynamic spindle depends on the remarkable properties of microtubule.
Please do not skip the ad.
Hotel Trivago.
Okay, here's a short recap of what we have learned just now.
Microtubule continuously polymerize and depolymerize by the addition and loss of their tubulin subunit, and individual filaments alternate between growing and shrinking, a process called dynamic instability. At the start of mitosis, the stability of microtubule decreased because MCDK phosphorylate microtubule-associated proteins that influence the stability of the microtubule. As a result, during prophase, rapid growing and shrinking microtubule extended in all directions of the two centromere, exploring in the interior of the cell.
Metaphase starts abruptly with the diassembly of nuclear membrane. Do you ever think what the process occurred during the breakdown? The best understood of this event is depolymerization of the nuclear lamina, which is the meshwork of filaments underlying the nuclear membrane. Active MCDK phosphorylates the main switches the protein that make up the nuclear lamina, phosphorylation leading the breakdown of the nuclear membrane. As the nuclear lamina dissociates, the nuclear membrane fragments into vesicle. After the breakdown, the spindle microtubule can gain access to the duplicated chromosome to start attach at the kinetochores. Kinetochore will recognize the special DNA sequence present in the centromere. Microtubules are attached to the kinetochore via interaction of multiple copies of an elongated connectin protein complex. This complex will bind to the +N microtubule, which allow microtubule to grow or shrink and attach to the kinetochore. Kinetochore and sister chromatic face in opposite direction. They tend to attach to microtubules from the opposite poles of the spindle. The attachment of opposite pole called biorientation. The tension formed as a result from the biorientation signals the sister chromatic that they are attached correctly and ready to separate. The cell cycle control system monitor this tension to ensure correct chromosome attachment. This event shows the end of the pro-metaphase.
Have you ever wondered what is the main structure for chromosome alignment? The main structures involved in the alignment of chromosomes are microtubules and kinetochore. On this subtopic, I will focus on the structure of kinetochore. Kinetochore is a complex structure that specifies the attachment between the chromosomes and the mitotic spindle that will hold the chromosome together. Kinetochore has a mechanical tension sensing system to monitor proper alignment and attachment of chromosome, and a stop signal broadcasting system to signal chromosome separation. The attachment of kinetochore and the mitotic spindle will generate tension forces created by the process of depolymerization. The cell cycle control system monitors tension forces to ensure all chromosomes attach to the mitotic spindles correctly. If the chromosomes are unattached, they will send a negative signal to the cell control system to inhibit activation of APC. Continual growth and shrinkage of microtubules are required for the arrangement of microtubules and the chromosomes into the bipolar spindle. The kinetochore are being pulled in the opposite direction towards the center of the cell until microtubule tension is balanced on both sides of the centromere. The mitotic spindle keep oscillating back and forth when the chromosomes gather at the equator to adjust their position until they form a metaphase plate. This event signaling the beginning of metaphase. Before entering the anaphase, the spindle assembly checkpoint will ensure all the chromosomes properly aligned at the metaphase plate to avoid having an incomplete chromosome set.
Summarize what we had learned throughout this video are: first, the centrosome is the principal microtubule organizing center. This is because the centrosome helps in the formation of two poles of the mitotic spindle. Next, the centrosome will form aster and separate. The aster form will move each of the centrosomes to the opposite sides of the nucleus, forming the two poles of mitotic spindle. During prophase, the MCDK will phosphorylate the microtubule associate proteins and influence the dynamic instability. Some of the microtubules will interact with microtubules from other centrosomes, joining together forming the mitotic spindle. Centrosomes are now called spindle poles, while the interacting microtubules are called interpolar microtubules. Next, during prometaphase, the spindle microtubule from the centrosome can access and capture the duplicated chromosomes. When nuclear envelope disassembled, the spindle microtubules will bind at each kinetochore at opposite sides of the chromosome. The attachment of spindle pole and the kinetochore called biorientation. This will generate tension on the kinetochores, give signals to the kinetochore to attach correctly and ready to separate. Lastly, during metaphase, the duplicated chromosome will align at the equator, halfway between the opposite spindle pole. The mitotic spindle that attached at the duplicated chromosome jerked to its opposite pole and aligned the chromosome at the equator. Thus, the metaphase plate is formed. The chromosome will then continually adjusting their position at the equator before separate in anaphase later. That is all from the video. Thank you.
This video is not sponsored by Trivago.